We'll see how it goes. We're not sure yet. You know, I need one of those Madonna things. You know, that comes around like that. Okay, are we good? If you can turn up the amp, you know, maybe a little bit, that'd be great. So this is, you know, a bunch of old friends are here, which are wonderful to see. There's also just enough chemistry graduate students on it, so a little intimidating. Also, before I start, I want to acknowledge women in science and engineering. I think it's really wonderful, Alicia and her colleagues, to put this on for our wonderful Santa Cruz community. A lot of them. Okay. So, so that's me. Oh, wait, I have my pointer here. Oh, and I have a beer. So I've never given a lecture with a beer. I've been doing this for like 25 years, and I got a beer. This is great. Okay. So, pardon me?
Follow the mic. We can't hear you.
You still can't hear me. Okay. I can't get any closer than this. So we're going to have to turn up the gain on this.
Yeah,
we're not getting
Alicia's
feedback,
gonna
so
play
I think
with it.
we turn up the gain. Put it up to 11. A few people go, oh, John knows how to do this stuff. Okay. Testing. Okay. All right. How's the gain? Okay. Is that a little better? All right. If I wander away, okay, so I'm trusting you people in the back. If I wander away, just, you know, wave at me. All right. Okay, so now what I have to do is be able to reach the computer. Okay, so mad cows, cannibals, neurodegenerative disease. Oh, and by the way, this is what a UCSC chemist looks like right here. Okay, so I'm actually going to break this talk out into a couple of little vignettes. And the first one, I'm going to tell you about a little bit of a mystery, okay? And truthfully, at this point, it's one that is not solved. And then I'm going to tell you a story, a historical perspective on what we kind of has led us to current understanding of certain types of neurodegenerative diseases. I'll talk about proteins. I'm going to be talking tonight a lot about proteins. And so I'll explain to you folks what proteins are and the different things, some of the different things that they do. I'll touch on just a little bit of our laboratory's work, and some of our graduate students are here. Kate, Jill, a few others. And then at the end, if we have time, one of the things I think often gets lost in scientific talks is actually who does the science and who pays for the science and what's it really like being in a scientific laboratory nowadays. And so we'll spend just a couple minutes on that. All right, so here's the mystery. All right, so now, as I said, I'm going to be talking about proteins. And proteins are molecules that our bodies use. They're made up of amino acids, and if you don't follow all the details at this point, don't worry, because I'll show you much, much better figures in a few minutes. But suffice to say, a protein is made up of a bunch of amino acids strung together. They all have different types of amino acids. Some are long, some are short, etc., etc. And oftentimes, what you want to do, so this is something I always do with school kids. So the question comes up, how big is a protein? Okay, so we're talking about molecules here. So molecules are small, okay? So what we have to do is, so you know that thing you do when you're a little kid and you put your fingers together and you see if you can get sunlight through them, okay? So hold your fingers up. We're not too old to do this, all right? So you hold your fingers up and you look at a light, okay? You press your fingers together as small as you can until the light barely comes through, okay? So at that point, that little pore that you've made in your fingers is about 1/10 of a millimeter, okay? A protein, a molecule, is 1,000,000 times smaller than that, okay? So that's the scale of the type of molecules I'll be talking about today. Very, very small, but very complex, as you'll see, as things go along. Okay, so here's the mystery. Now, there's a type of experiment, we don't do this, but a number of our collaborators do, where if you discover a protein, so our bodies, our genome, in a sense, the DNA that we have, codes for about 10,000 different types of proteins. They all carry out different functions. Some are for structure, some are for digesting the food you're eating, some are for breaking down the alcohols you're taking in. You know, many, many different things. They transmit excitability along nerves and so forth, okay? If you discover a protein based on its sequence and you don't know what that protein does, what oftentimes is done as an experiment to find that out is you develop what's called a transgenic mouse. So this is a mouse. It's a laboratory technique in a sense where you knock out the gene for that protein and you see what happens, okay? You see what function is lost. So here's the mystery, okay? So there's a protein. We'll give it a name right now. We'll call it PRP. That stands for prion protein. I'll tell you all about that in a few minutes. So this is the prion protein right here, okay, and we'll just let the little dots represent amino acids, okay? So the animal has the prion protein. If it has the normal prion protein, that's called a wild type, okay? That's a standard mouse that has the regular protein. Now, you don't know what that protein does, so you knock out, so this is what the empty circles are meant to represent. You eliminate half of the protein. And the N and the C are just kind of a naming that we protein chemists use to measure or mark the two ends of a protein. Okay, they have distinct chemical features. Okay, so you knock off that piece on the N-terminal side, and that animal is completely fine, no effect whatsoever. So you say, all right, well, let's take this a little bit further. Let's eliminate the entire protein from the animal. Okay, it completely does not have that protein anymore. That animal is also completely fine. Okay. Now, through a series of almost accidental experiments, and this took place about 20 years ago, some scientists eliminated, you see the little movie there? Okay, right? Eliminated just a few amino acids in the middle, and that animal dies. Okay? And as a matter of fact, what happens is the neonates of that animal are born, okay, into a litter, and they die within a few days. So that animal can survive without the protein. Of course, it survives just fine with the protein, okay? But you knock out this teeny little segment in the middle, and somehow that's so toxic that the animal cannot survive. So how do you explain this? And I'll tell you right now, I'll give you an idea at the sense, but in general, the field does not understand this. And in the field of neurodegeneration, which I'll be talking about, this is one of the major issues that scientists are trying to solve. All right, so that's the mystery. So now for some history, okay? So the current understanding of the type of diseases, the specific type of neurodegenerative disease I'm going to be talking about tonight, actually began back in the early part of the last century, okay? And in Papua New Guinea, this part of the world over here, okay? And of course, this, with the tragedy of that jet, this part of the world is getting a lot of attention right now. But this is in Papua New Guinea. There was a disease being spread among tribal folks. And these tribes were known as, and they still exist today, as the Fore people. It's just spelled F-O-R-E, but it's pronounced Fore, okay? And tribal folks, very, very primitive. They still are quite primitive to this day, okay? And back in the early part of the last century, especially through the 1940s, 1950s, there was a horrible, in a sense, infectious disease spreading among the Fore people. And it was taking tribal members out in very, very large amounts. And in fact, it was so prevalent that it was getting worldwide attention. The whole biomedical community at the time was paying a lot of attention and trying to follow this. Now, interestingly, so here's a young fellow who's suffering from the disease. Oh, so I didn't mention. So they were suffering from this neurodegenerative disease called Kuru. Okay, and this disease was spreading among the Fore people. Okay, so Kuru in the Fore language means to shake. And the way this disease would develop is initially individuals would lose coordination of their hands and legs. They would have a difficult time walking, okay? Eventually they lost the ability to walk. They would have a hard time swallowing, have a hard time breathing. There would be cognitive dysfunction, and eventually they would succumb and pass on. So this disease, Kuru, as I was saying, was spreading like crazy among the Fore tribal people. And it was unclear as to what was going on. So this is a young fellow who's suffering from Kuru over here. And this is showing a number of additional folks who also had Kuru at the time. And I should mention this disease was always fatal. And interestingly, this disease was transmitted primarily among women and children of the Fore tribes. Okay? So the question is, what was going on? So there was a really phenomenal scientist, physician at the time, named Carlton Gajdusek. And Gajdusek is shown right there. And this captured his interest, and he went to live among the Fore people to try to study this disease and figure out what was going on. Now, of course, you could tell from the title of the talk, Mad Cows, Cannibals, one of the things that was very interesting about these people is that they were cannibals, okay? They were very, very short in protein sources in their diet. And so part of their culture, their ritualism, is that when a family member, a loved one would pass on, they would carry out a so-called mortuary feast where they would consume the tissues of that individual, okay? Yeah, I know, June's old friend of mine over there is...
Grimacing.
Yes, wincing, yes. And I can point you in the direction of books that go through lots of the details of this. So, interestingly, as I mentioned, this was passed primarily among women and children. So in the mortuary feast, what would happen is these were very sexist societies, okay? So when they were carrying out the mortuary feast, the cannibalistic rituals, the men would come in and they would make the choices that they wanted for their meal, for the cannibalistic meal, okay? And of course they would make the same choice we would all make, and this isn't a great discussion for a meal. I apologize, those of you. But they would take the muscle cuts, okay? And they would leave the nerve tissue to the women and the children, okay? And of course that's what was propagating the disease. Whoops. I'm getting too close to this microphone. Okay. So Biochemistry reasoned that there must be something infectious in the nervous tissue that's being passed along through these cannibalistic rites, okay, and that's what's going on. Now, of course, that is what was transmitting the disease, but Biochemistry, being the phenomenal scientist that he was, took this a lot further and actually set up a laboratory in the jungle and started analyzing the tissue and sending samples back to the National Institutes of Health and other scientific collaborators to try to understand what was going on. So now you can see, this is a normal brain slice over here. This is diseased, okay, and it looks the same. I'll show you there's actually more modern diseases, but a Kuru brain, okay, shows these types of openings, okay, this so-called vacuolization, okay, and it basically makes the brain look like sponge, okay. So since these diseases are infectious, and since it turns the brain into sponge, okay, you have to give it a very, very fancy, complicated medical name, okay? So these are called transmissible spongiform encephalopathies, okay? And nobody ever says that. You say it once, and then after that you call them the TSEs, okay? And so anyhow, you can see what's going on. But there was additional features, okay? So now, we're all familiar with infectious disease, right? So Kuru is an infectious disease, okay? It's passed on by consuming brain tissue, all right? And when we think of infectious disease, all of us in modern 21st century society think of one of two things, right? Virus or bacteria. Something that has a genome, something that can replicate, okay? You get a cold, you get flu, pneumonia, whatever it is. There's always some microorganism that's propagating this disease, all right? So Gajdusek took the Kuru tissue, okay, and cultured it. If there are bacteria or virus in there, you can put it in certain media and grow these organisms up, get enough of them, and then start analyzing for what they are. Well, try as they might, they never found any virus or bacteria at all whatsoever, okay? And they did not exist. And as a matter of fact, you can even go a step further and treat the tissue in a way that would eliminate all the viral or bacterial infectivity, and the tissue from inoculation experiments would still be fully infectious. So there was something else going on, okay? Now what you're not seeing here in these micrographs is, okay, in these slices over here, is there was an accumulation of something, and it wasn't virus or bacterium, it was protein, okay? The Kuru brain is loaded up with a certain type of protein, and I'll show you in a minute or two what that protein is. So these diseases exist today. So fortunately, based on Gajdusek's recommendations, cannibalism stopped, okay? It's no longer practiced. The 4A people still are very primitive, living their life in Papua New Guinea. But fortunately, Kuru has all been eradicated. But these diseases still exist, and they exist in a number of animals as well as humans, as I'll show you in a second, okay? So, and now you'll notice in every animal I show you, the disease has a different name, okay? This maximizes the confusion, component, okay? Keeps out the uninitiated, okay? So in goats and sheep, it's called scraping, okay? We don't have scraping in the United States, but this is a problem in certain regions of Europe. And what the sheep do is they rub up against fence posts and so forth. Apparently their skin itches like crazy. And they scrape and they scrape and they scrape, okay, until they rub off all the wool, as you can see over there, okay? If you look at the brains post-mortem of a scrapey sheep, it looks just like what you see in Kuru, okay? The same sort of spongiform degeneration. In the United States, we have a huge problem. Now, we're not so aware of it in California because it's not out here, but in the Rocky Mountain states and up through Canada, there's a disease called chronic wasting disease, and this is taking out something like 20 to 30% of all the deer and elk in the wild. And it's a horrible agricultural problem, and as much as the federal government has tried to eradicate this by by culling out sick animals, moving healthy animals to different regions and so forth, they have not been able to eradicate it. If anybody's interested later, we can discuss why. And of course, there's mad cow disease, okay? Mad cow disease does not exist in the United States. Every once in a while, you'll hear about a mad cow in the press, and then everybody stops buying meat, and it's a catastrophe for the meat packing industry, the meat industry. But as many of you know, in the late 1980s, early 1990s, this was a huge agricultural problem in the United Kingdom. And there were many diseased animals, and hundreds of thousands had to be sacrificed and eliminated. Now, you might be wondering, okay, so the 4A people were cannibals, and they were passing along this disease by eating diseased nerve tissue that had this accumulated protein, right? Okay, but anybody that knows anything about cows knows that they're herbivores, right? So how the heck was this disease being amplified in them to the point where hundreds of thousands of animals were getting sick, okay? Well, one of the things that we don't know, or is kept from us, okay, is that in slaughterhouses, there's no such thing as waste material, okay? There's so many animals going through the slaughterhouses that every, cubic millimeter of tissue has to be taken care of. So anything that can't be sold, okay, is scraped off, rendered, repackaged, put into feed, and fed back to cows, okay? So the meat industry was actually forcing the cows to be cannibals, okay? And that's what amplified the disease, and we don't allow that anymore, and mad cow disease is essentially gone. Okay, it exists in humans also, okay? And in humans, there's actually several different diseases beyond Kuru, but the most prevalent is called Creutzfeldt-Jakob disease, and it always has these horrible long German names, okay? But interestingly, Creutzfeldt and Jakob, two individuals, were contemporaries of Alzheimer, okay? And they were comparing notes back about 100 years ago at the turn of the last century, trying to understand these various plaques and degeneration that they were observing in different types of human diseases. So Creutzfeldt-Jakob disease, very rare, thank goodness, okay? About 1 in a million deaths in the United States are from CJD, Creutzfeldt-Jakob disease. It's kind of like Alzheimer's disease, but a much more rapidly progressing Alzheimer's disease. You can think of it as Alzheimer's on steroids. It goes very rapidly. In addition, whereas Alzheimer's disease is strictly a cognitive disease, okay, it leads to dysfunction of the brain, CJD actually leads to loss of coordination as well. Okay, so there's a differential diagnosis going on there. Now, it's typically like Alzheimer's disease, a disease associated with aging. When it kicks in a sporadic fashion, meaning that there's no infectious route, it takes place later in life, 60s, 70s, 80s. But there is a form of the disease called variant Creutzfeldt-Jakob disease, which this poor individual died from over here, and she contracted that by eating infected mad cow tissue back in the 1990s, okay? So fortunately, this is now all out of the food chain, but there were a number of individuals. So I didn't bring along any charts, but only about 200 people. There's a huge concern a number of years ago that there would be literally thousands, maybe tens, hundreds of thousands of individuals dying of this. And it's been unwatched, and there's no reason why, in principle, that shouldn't have happened. Thank goodness it did not happen. Only about 200 individuals so far have died of variant Creutzfeldt-Jakob disease. Okay, so now here are protein plaques, all right? So as I've told you, this disease somehow is linked to an accumulation of a protein. So this is what a plaque looks like. This is a higher magnification micrograph compared to what I showed you before. And if you can see, it's a little bit small, but that region right there, that's the protein plaque of Creutzfeldt-Jakob disease. And it sort of has a flowery look to it if you get close to it. So it's actually called a florid plaque, all right? Here's another plaque over here, taking on sort of a different look. Yet another one over here, okay? But interestingly, those are not from this type of disease. This is not Creutzfeldt-Jakob disease. The one in the middle is Alzheimer's disease, and the one on the right is Parkinson's disease. So all of these diseases are highly related to each other in the sense that they involve accumulation of protein in the brain and subsequent neurodegeneration. Okay? Does that make sense so far? Yeah? Okay, great. Plaque. Okay, so a plaque is, and I'll show you a better image in a second. A plaque is an accumulation, okay, something that the body cannot eradicate, and it's a collection of protein, okay, that's all sort of come together. And I'll show you something a little bit higher resolution in a minute, okay? But it's interesting because, in a sense, the plaque itself, comes in many different forms, okay? So it's an interesting point. All right, and this gets to your question. So what, in a sense, are these plaques? Well, what happens is these are rich in protein, and proteins, if they take on, in a sense, the wrong shape, and I'll show you what I mean by that in a second, they can actually start sticking to each other and start forming little, what we call fibrils, okay? And in the brain, Alzheimer's disease, Parkinson's disease, prion diseases, CJD and so forth, you get these long-forming protein strands. These are not natural. We don't have these naturally, but these are always associated with these diseases. So, and I should mention, these are often referred to as amyloid plaques. and this is a really archaic name. Now, the chemists in the audience, I should quiz you guys, okay? But AMYL, okay, amyl, okay, in chemist language means sugar, okay? So even though now we know it's protein, 100 years ago when these were first discovered, they had the properties that made early chemists think they were sugar-like. So they referred to these as amyloid, which means sugar-like, but it has nothing to do with sugar whatsoever, it's protein. Okay, so these are amyloid plaques. So, so we all know that chemists are the most badass people around. So this is from Breaking Bad, for those of you who are not TV watchers. So I love this, you know. So chemists are thought of as perhaps, maybe along with physicists like my old friend George over here, you know, very mild-mannered people. But I love the fact that the most badass guy on the best show in the history of TV was a chemist. Okay? And it's because we can make things, you know. We're dangerous. You want to keep us happy. Okay, but actually, more seriously, one of the magical things, one of the really wonderful things about chemistry is that chemists are really, really good at representing molecules in lots of different ways. The world is molecules, okay? It all comes from the periodic table. The periodic table is the palette of our world, okay? It's the palette of life. Everything that makes us up is drawn from those less than 100 elements that are actually relevant, okay? Yeah. And those are packaged together in different molecules. And chemists they learn a special skill to represent molecules in a way that puts things in the context that you need, depending on what it is you're trying to understand. So those of you that are not chemists probably recognize that each one of these things kind of looks molecular, okay? They all look really different, right? Okay? So obviously, this is a little puffball representation, technically the puffball representation, okay? So this is an amino acid over here, and every atom is represented as a little sphere, and probably at some level they kind of look that way. Over here, you obviously have the double helix of DNA, but you don't see any of those spheres. But this captures the shape of DNA, which at this particular level is what would be important, okay? Similarly, here you don't see any puffballs, you see lines and letters, right? This is yet a different type of representation, and you use different representations depending on what it is you're trying to understand, okay? And so let me tell you what some of these things are. So let's see. This is a hormone called MSH, melanocyte stimulating hormone, and this actually suppresses your appetite. So if you guys are full, I notice that most of you have stopped eating. That hormone level in your body is now upregulated. Okay. Let's see. This is ethanol. So if you're drinking beer, wine, this is the alcohol. Okay. This is caffeine. Later, you know, before you drive home, this will be in your cappuccino, and so forth. And that's an amino acid called alanine. So all of these are different ways of representing molecules. So with proteins, we need a special way of representing them. So I want to give you a little 1-minute class on how we represent proteins. So if I showed you a protein, so proteins are bunches of amino acids all linked together, okay? And if I show you a protein and the way it probably really looks as a molecule, with each atom is a little puffball, it kind of looks like that. And it conveys absolutely no information whatsoever. There's not a chemist in the world, no matter how long they've been doing it, that can look at that and make any sense out of it, right? Okay? So you think to yourself, well, okay, let's get rid of the puffballs and let's turn it into sticks. Maybe it'll look a little bit better. So we turn it into sticks, and it looks like that, okay? And it looks a little bit better. You can kind of see some of the connectivity up here, and you can see some side chains sticking out, but it still is pretty ridiculous, okay? It's not the kind of thing that's terribly useful if you want to know something about the shape of this protein. So you go a little bit further, and you do that. And now it looks sensible, doesn't it? So this is called a ribbon diagram. This is how protein chemists like to represent proteins. So we eliminate all the details of the amino acids, and what we do is we just show how they're connected to each other. And we trace along, so you have, the way you think of a polymer, a protein, is amino acid, amino acid, amino acid, amino acid, okay? There's 20 different types of amino acids. The unique protein will have one type of amino acid here, another one there, etc. What makes one protein different from another is the order of the amino acids and how long the protein is and so forth. And so what this representation is showing is just how they're connected to each other without any identification of the specific amino acid, okay? All right, so that is so-called ribbon diagram. And this sets it up so that we can now start understanding what different proteins do. So this just shows you a range of different proteins, different shapes, and it shows you sort of the library, a menagerie of just a few of the 10,000 different proteins that our bodies produce, okay? So there you've got a helix, and I should mention that corkscrew thing, that's called an alpha helix. That structure was actually predicted by Linus Pauling, okay, back in the 1940s, I believe, okay? And you can see, and you have different things showing up over there. All right, so proteins have shape, so that's the idea. All right, so now how do you develop one of these protein-based diseases, and how can a disease like this be infectious? Okay, this is the issue, all right? So what we want to imagine, and this is really the way one thinks about it, okay, is that we've got a protein, and we're going to give it a name. We're going to call it the prion protein. So the protein that causes mad cow disease, scrapie, Creutzfeldt-Jakob disease is named the prion protein, okay? And it's that same protein I was showing you at the beginning, the mystery, okay? Now that protein can have a couple of different shapes, okay? And we'll give those shapes names. So one we'll call a PRPC. That's good, okay? And all of you have this protein, okay? All mammals have this protein, okay? Your cat has this protein, okay? So this protein is ubiquitous, okay? And there's a lot of it, all right? So the good form is called PRPC. The C stands for cellular, okay? Now somehow that protein can take an improper so-called fold, a different shape. That's the bad one, and that's called PRPSC, and that stands for scrapie, so the scrapie form of the protein, okay? So now what you imagine, so I'm a chemist, and this is a chemist's view of a biological cell, okay? So, it's round, and there's, that's how it's good enough for us, okay? So the prion protein decorates the outside of nerve cells. Of course, they're much, much more complicated, but it decorates the outside of nerve cells, all right? As I say, we all have it. So this is a cell. Now, and I should mention, this will be the cheesiest movie you've ever seen in your life. It's absolutely terrible, but it works. My postdoc in the lab made this years ago, and I like it. Okay. So now along comes a bad form of the protein, okay? So this protein now comes in, either it forms spontaneously, sporadic disease, or it comes from infectious tissue that somehow is consumed, it gets into the central nervous system, okay? And along it comes and it docks to a healthy form of the protein, okay? Now there's something very stable, as we like to say in the chemistry world, about that misfolded form, and when those two come in contact with each other, somehow the red guy convinces the blue guy to become another copy of the red guy, okay? So this is a problem in geometric combinatorics, right? If 1 goes to 2, and 2 can do this, 2 becomes 4, 4 becomes 8, etc., etc. And this in chemistry is what we call autocatalysis. So this grows and grows and grows, and eventually it starts overtaking the cell. We change the color of the membrane, okay, and the cell breaks up and it dies, okay? So that is kind of what's going on. We're basically, by having this misshapen protein, it's causing other copies of the protein to take on this improper fold, and that then spreads and spreads and spreads, and that leads to the notion of the plaque, okay? So this now is a cartoon representation of the plaque. All right, so now, all of these diseases come about the same way, okay? Alzheimer's disease, Parkinson's disease, the prion diseases, they all come about from different proteins, all right? So, prion diseases, from the prion protein, that was that ribbon diagram I was showing you before, we refer to that as PRP. Alzheimer's disease comes from a different peptide, okay? It's just a short protein called A-beta, okay? The amyloid beta peptide or protein. Parkinson's disease comes from another protein called alpha-synuclein, all right? All from different proteins. So when I think about and when I phrase sort of neurodegenerative disease, I always pose it in terms of 3 questions, okay? Number 1, why do these specific proteins form plaques? So like I was saying before, our bodies have 10,000 different types of proteins. What is it about these that makes them form plaques? Okay, number 1. Number 2, why are the plaques toxic to brain tissue? These are molecules produced by our body, but somehow when they form these plaques, it causes this horrible neurodegeneration, okay? And number 3, what is the function of the precursor protein, which is just a chemistry way of saying, why the hell do we have it, okay? Why do we have the prion protein? Why do we have the Alzheimer's A-beta peptide, okay? And so forth. So, no answers, okay? Was that dramatic enough? Okay. Okay. No answers, all right? So this is why, so part of the title of the talk is why is it so difficult to cure Alzheimer's disease? And it's because one doesn't even really know where to start, okay? This is a very, very challenging thing compared to other diseases, and I'll draw some parallels in a second. All right. Now, what is, okay, so one of the things, and this now gets into a little bit of what our laboratory does, and I'll just talk for a couple minutes on this, but one of the things that we believe is that if we can understand something about these neurodegenerative proteins, okay, perhaps we can figure out what function is being lost when they take on that improper shape, okay? So one of the things that we now know about the prion protein is that it's intimately involved with metals in the brain, okay? So we always like to think about biological molecules, we think about sugars and fats and, and proteins and so forth, okay? But in fact, we can't live without metal ions, okay? And primarily copper, zinc, and iron, and the brain is absolutely rich with these metal ions. So everybody knows the story, right, of iron in blood, okay? So you take a breath, okay, you breathe in oxygen, you breathe in air, the oxygen diffuses across the alveolar membranes, okay? It binds to hemoglobin in your red blood cells, that's transmitted throughout the body. When the hemoglobin blood cells get to low oxygenated tissues, it releases the oxygen and so forth. That's great, okay? But now the oxygen is at the cells and you gotta be able to use it. Well, you can't use it unless you have copper, okay? Copper is absolutely essential for cellular respiration through a series of proteins called oxidases, okay? Something we won't get into, but suffice to say the brain has a very, very high metabolic rate. Matter of fact, it has higher cellular respiration than your muscles do, even when you're exercising like crazy. The brain is constantly using oxygen, constantly going through cellular respiration. And it absolutely needs copper, iron, and zinc. Okay, and I won't go into the roles of all these different things. So we now know, based on a lot of data from our lab and other laboratories, that the prion protein is really, really important for regulating these metal ions. So, copper, good or bad? So when we think about copper, if somebody mentions copper to you, you think of a penny, right? Coinage metals, okay? Or you think about copper wire. I know that's what John Cohen thinks of, wiring things, okay? Rain gutters, bathtubs, whatever. This is not what a chemist thinks about. When a chemist thinks about copper, they think about that, especially a biochemist. One atom of copper, okay, stuck in the middle of a protein somewhere, there to carry out a very, very specific biological function, okay? And this is what the prion protein does, okay? It binds metal ions in a way that's very, very important for neurological function. Okay, so how do we do this? We have a bunch of fancy instruments at UC Santa Cruz. If anybody ever wants to come up and take a tour, send me an email. I'll be happy to tour you around the lab. These are called electron paramagnetic resonance spectrometers. I won't say anything more about them other than the fact that if you've ever had an MRI, this is kind of like MRI but at the molecular level, okay? And it images what's going on around metal ions. Okay, so now this is what we believe the prion protein actually looks like. So this is now showing the prion protein in all of its glory, okay? So here is a cellular membrane. This is a model based on data from our lab and a number of collaborators' labs and so forth. This is the ribbon region that I was showing you before in that rendering, okay? It's stuck to membranes through this chemical group right there. It's called a GPI anchor, which has this incredibly long, horrible name that I won't even try, okay? It has sugars bound to it, okay? So it's actually a so-called glycoprotein, and these carbohydrates are very, very important, okay? And then these blue spheres are actually copper ions, okay? Prion protein binds these different copper ions, and in doing so, it facilitates the movement and the utility of copper in the brain. All right, now, a very bright former graduate student in the lab, Dan Spevacek, Dan's now a postdoc at UC Davis, was suspicious about the shape of the protein. So what I showed you before is, here's what we call the C-terminal region, and here's the N-terminal region. They look like they're kind of separated from each other, right? You have metal binding over here, you have this ribbony region over here, okay? And she was suspicious, based on a lot of biological experiments from other laboratories, that in fact those two regions of the protein were somehow interacting. And she carried out a whole bunch of chemical experiments, immunogenesis experiments, magnetic resonance experiments, and put together this really beautiful story that showed, in fact, and this is showing just one metal ion, this is actually zinc, and instead of copper, it interacts with both metal ions, that in fact these two regions of the protein are intimately connected to each other, okay? And even though it looks like just a ribbon picture over here, it turns out understanding this has taken us a long way down the road towards understanding certain types of prion diseases, certain diseases that arise from this protein. Okay, now do you remember at the beginning when I told you about the mystery, okay, that you could eliminate the prion protein, okay, and the animal would be okay. You could eliminate the N-terminal region of the prion protein, the animal would be okay. But you eliminate that middle region and the animal dies, right? Okay, can you guess where that middle region is? Duh, okay, highlighted in red, okay. So that region is very susceptible to a type of chemical reaction